In this talk, I'm going to discuss a comparison between skin disease classification model explanations and human intuition about the important factors for doing this type of classification. There's been significant previous interest in skin disease classification from consumer-grade camera images. This is a challenging task for several reasons, but one of the biggest is heterogeneity in image content. As you can see from the example images here, skin lesions present in varying shapes and sizes, manifest on different body parts, and present differently in patients of different sex, skin tone, and age. Also, consumer-grade camera images of skin lesions contain widely varying background content and markings, like the office furniture and the ruler in these photos. For that reason, it's important to know whether a skin classification model is using the pathology visible in the image to make classifications, or whether it's using other features. There already exist tools to generate explanations of input features important for model classifications for a single image. In this talk, I'm going to show you how we quantitatively compare these explanations with human intuition about important features for diagnosis. So, for example, when you look at the image on the bottom right of this slide, you might expect that characteristics of the lesion circled in yellow are important for making a diagnosis. We want to understand how that type of intuition agrees or disagrees with what models are using to generate classifications. The methods I'm going to describe in the rest of this talk allow us to identify trends in that agreement across the entire dataset, instead of observations about a single image. Next, I'll walk through the pipeline for computing the agreement between model explanations and human intuitions. So, we start off with a large dataset of images used to develop the skin disease classifier. This dataset comprised about 20,000 adult cases from a teledermatology service. For each case, we had between 1 and 6 images, plus some metadata describing general information such as the sex and the age of the patient. A group of board-certified dermatologists labeled each of these cases into one of 26 conditions or an other category. More details about that dataset are available in the paper, A Deep Learning System for Differential Diagnosis of Skin Diseases. For the purposes of this work, we selected images from a subset of about 1,300 of those cases. The cases in this saliency evaluation dataset were drawn from the same distribution of conditions as in the larger dataset used for model training. For each image within the saliency evaluation dataset, we computed a human-labeled region of interest. In particular, for each image, 3 dermatologist-trained graders labeled regions of interest for diagnosing the skin condition present in that image. We overlaid each of these labels and computed the Fleiss-Kappa score to measure inter-rater agreement on these labels. In general, most of the labels showed high agreement. We discarded images with a Fleiss-Kappa score below 0.4. Amongst the images with high agreement, we took a pixel-wise majority vote to determine the final human-labeled region of interest. For each image, we also computed a model explanation describing the important input features for a skin condition classifier. The classifier we used is a neural network presented in the paper, A Deep Learning System for Differential Diagnosis of Skin Diseases. That model has several arms based on an Inception v4 architecture, which takes in images. The metadata for each case is concatenated at the pre-logit layer. This model achieved a top 1 accuracy of 66% on 27 conditions, which is superior to primary care physicians and on par with dermatologists. We explained the predictions made by the model using a saliency map, which assigns a score to each pixel in an input image representing how important that pixel was for the model's final classification. In particular, we used a method called integrated gradients to generate our saliency maps. Integrated gradients computes the average derivative of the network output score with respect to each pixel location for several images along a path from a baseline x prime, in our case, a blank image, to the input image x being analyzed. More information about the integrated gradients technique is available in the paper, Axiomatic Attribution for Deep Networks. We also internally performed experiments with the GradCam attribution technique, which yielded similar results as integrated gradients. After producing the saliency map, we thresholded it to generate a segmentation of the saliency map. That segmentation was compared to the majority consensus human-labeled region of interest via a dice score, which is a commonly used similarity metric for 2 regions. To perform another comparison that does not depend on a selected threshold, we also computed the Spearman rank correlation between the saliency map itself and the human-labeled consensus region of interest. The Spearman rank correlation computes the similarity between the rankings of important pixels in the saliency map and in the human-labeled region of interest. So, at the end of this process, we had 2 metrics analyzing the agreement between human-labeled majority consensus regions of interest and the saliency map for each image. Of course, for each case, there are multiple images, and we computed a Spearman rank correlation and a dice score for each one. We summarized each case by the single image with the highest Spearman rank correlation and the single image with the highest thresholded dice score. Of course, these were often the same image. This procedure took into account cases where only one or a few images contained content sufficient for diagnosis, so the model was not penalized for ignoring some less informative images in those cases. We then sorted all of the images in the saliency evaluation dataset by both the thresholded dice score and the Spearman rank correlation. The images on this slide are images from cases that ranked in the top 10% of the dataset in terms of both metrics. Qualitatively, we see there is indeed good agreement between the saliency maps in green and the human-labeled regions of interest outlined in yellow. These results suggest that the model is using information from the correct regions of these images to perform classification. For the incorrectly classified images on the right, this suggests that the misclassification is not based on mislocalization, but instead on an incorrect interpretation of the accurately identified lesion. On the other hand, here are images from cases that ranked in the bottom 10% of the dataset in terms of both metrics, and qualitatively, we can confirm that there is a disparity between the saliency maps and human-labeled regions of interest. For the correctly classified images on the left, further investigation is needed into how the model was able to correctly classify these cases despite low agreement. One possibility is that the model identified characteristics that are correlated with certain conditions. For example, white hair might be correlated with androgenetic alopecia, the condition in the bottom left image. For the incorrectly classified images on the right, the model's misclassification can be explained by the fact that it focused on regions of the image that are not related to the pathology being examined. Across all of the examples on this slide, and more generally across the lowest agreement examples in the dataset, these images tended to contain nails, hair, and lips. Interestingly, the lowest ranked examples did not tend to highlight background image features that might have distracted the model. We further analyzed these metrics stratified by skin condition. This scatter plot shows the mean Spearman rank correlation plotted against the mean dice score for correctly classified examples of conditions in green and incorrectly classified examples of conditions in red. First, we notice that categories with a higher Spearman rank correlation tend to also have a higher dice score, suggesting that these two metrics are consistent with each other. Inspecting the correctly classified examples, two of the conditions with the lowest scores are androgenetic alopecia and acne, with sample images shown here. These images tend to involve the hair and the face, consistent with the previous qualitative findings. Next, inspecting the incorrectly classified examples, two of the conditions with the lowest scores are seborrheic keratosis and melanoma. For many of these images, the model again tends to assign high importance to normal anatomy, such as fingernails. Finally, we stratified our analysis by demographics and performed statistical significance testing on whether differences existed based on sex, skin tone, and age. We observed a statistically significant difference based on sex, but not based on the other two characteristics. In summary, we presented a technique to quantitatively compare model explanations to human-labeled regions of interest. In many cases, with the worst agreement between these two, the model explanations highlighted normal anatomy such as fingernails, hair, and lips, unrelated to the actual lesion that was present in the image. These results suggest that future work to expose the model to more examples of normal anatomy could be beneficial for model performance and for generalization across varied examples. More generally, the workflow described in this talk could be used to identify differences between model explanations and human regions of interest for any model. If you're interested in more detailed resources describing the skin classification data, model, and the other research related to what I presented in this talk, you can learn more about those from the papers listed here.